Another type of epithelial tissue is transitional epithelium. Transitional epithelium tolerates repeated cycles of stretching and recoiling and returns to its previous shape without damage. To note, stretching and recoiling, that's important there. That's going to help it change the appearance as stretching occurs. So when you look at transitional epithelium with a tissue, it's going to look differently at different points of the stretching. For instance, you're going to find transitional epithelium in the bladder, and the bladder is going to stretch differently throughout the day. So when you look at the bladder tissue, when the bladder's full versus empty, it's going to look different. So the fact that it can stretch makes it transitional. This picture does a good example explaining that idea. So here you have the bladder in the pelvis. You have an empty bladder on the top here. So the empty bladder is not stretched out, so these cells look like they're big and round, I guess. They look full. And then you can see the full bladder, the cells are going to be stretched out, so they're going to look a little more squamous. So I guess one way to see the empty bladder, these cells look more cuboidal, whereas the full bladder, these cells look a little bit more squamous. This picture here that I'm outlining right now, that's a classic example of what transitional epithelial tissue is going to look like under the microscope or on a quiz. It just kind of looks globular, which means, some of the cells look spherical, circular. Some of them, they're just all sorts of shapes, and the nuclei are all over the place. Also note the basement membrane, and then we have the apical surface on the tops of them. That brings us to columnar epithelium. There's 3 types we have here. We have simple columnar, pseudostratified columnar, and stratified columnar. The simple columnar absorbs and secretes. Pseudostratified columnar has cilia movement. And the stratified columnar protects. So we'll go through those here following this slide. All right, go ahead and add to your notes the information on here about simple columnar epithelium. The locations are the lining of the stomach, intestine, gallbladder, uterine tubes, and collecting ducts of the kidneys. Functions: protection, secretion, and absorption. When you look at the picture of the simple columnar, notice the nuclei are pretty much all in a row. You also have, depending on where it is in the body, like this must be an intestinal lining. This has microvilli on the top. These guys on there. If it's in other places, might have cilia on there. I take that back. Not cilia. That's a different one. So the microvilli help with absorption of materials. We'll talk about that more later, but helps to increase the surface area so you can absorb more efficiently. You also notice the good basement membrane, and then you have the apical surface on the top. All right, the pseudostratified ciliated columnar epithelium. This is where the cilia is located versus the microvilli. Cilia is more of a hair-like projection. Microvilli is actual tissue. So pseudostratified, if we're talking more about etymology here, pseudo means fake or kind of not really. So it's sort of stratified, but it's sort of not. When you look at this type of tissue, you have some columnar looking cells and you have some cuboidal looking cells, and they kind of squeeze themselves in there. Because they squeeze themselves in there, they're kind of stratified, kind of simple. but you'll notice the nuclei are all over here. They're not linear. And the cells themselves just take funny shapes. That's one way to look at it, and then looking at the cilia. Places where you'll find pseudostratified epithelium, the lining of the nasal cavity, in the trachea, in the bronchi. So those will be part of the respiratory system. Those are all lined with cilia to help to sweep away debris so it doesn't get into the body. It's also portions of the male reproductive tract. So functions would be to protect secretion, like secrete mucus, and then to move that mucus away and other stuff with the cilia. Okay, stratified columnar is true stratified. You can see distinct layers of cuboidal cells, and then on top of that you have a distinct single layer of columnar cells. You'll notice the nuclei in the columnar cells are lined up nice and straight, and all the cells seem to be stacked nicely next to each other. They don't seem squished like the pseudostratified. And then there's one or two layers of cuboidal underneath. So here you have the lumen or the apical surface, and then you have the basement membrane below. Locations of stratified columnar epithelium include the pharynx, epiglottis, anus, mammary glands, salivary glands, and the urethra. The pharynx would be your throat. The epiglottis is a little, kind of like a valve, if you will, in the back of your throat that opens and closes when you swallow, so you don't get food going down your trachea or your windpipe. And the urethra is the end of the urinary tract. So functions of stratified columnar will be protection. So those layers can help protect and replace themselves if they need to. Alright, as I go over this slide, I'll highlight what you need to write down. There's some extra information that we're not going to cover in this class, but this is your glandular epithelium. And there's several different types of glands. There's endocrine glands and exocrine glands. Endocrine glands release hormones, whereas exocrine glands produce secretions. Real quickly, we'll go over modes of secretion. There's three types: merocrine secretion, apocrine secretion, and holocrine secretion. We'll go over these in more detail next. Okay, in merocrine secretion, don't worry about the Golgi apparatus. When it produces the secretions, they're released by vesicles via exocytosis. So an entire vesicle will release out of the cell, and then it'll spew its contents out of the cell. So let me try my hand here at drawing. So we have your cell, you have your vesicle, and it's got something in it, and then it's for exocytosis, it's when the vesicle gets closer to the membrane, and it's just going to release the contents out. So that's merocrine secretion, and that's how sweat is released. So it goes through the glands. It's actual contents from vesicles. So sweat was in the vesicles and gets released that way. With apocrine secretion, you're actually going to shed the cytoplasm. So the cytoplasm is the secretion. So for example, with mammary glands, the milk produced actually is cytoplasm from the cell. So the cell itself, I want to probably have pictures here next, but you need to watch me draw. The cell itself will release open, and the cytoplasm comes out, and that goes to the ducts and goes out for the milk to be released. So that's apocrine secretion. And then finally, the holocrine secretion. Holocrine, the root word's like whole, is the whole cell bursts, and that cell is what comes out. And that's sebaceous glands, that's your oil glands. If you think about that, those glands, the oil kind of gets clogged up and there's a bunch of dead skin cells in there. So the oil itself, it comes from the cell bursting, so it's contents of the cell. That's holocrine secretion. The whole cell gets secreted basically out of the duct for like your oil ducts. Alright, this slide shows you better pictures than what I drew for you. Merocrine secretion, you have these little vesicles, you see them going towards the cell membrane, and they're releasing their contents out of the cell. So up here, you see the vesicles releasing the contents there. That looks like it's saliva in the salivary gland. And then apocrine secretion, you have the mammary gland here, so you have the cells. In the mammary glands, and they're going to secrete the cytoplasm. So if you read here, it says apocrine secretion involves the loss of apical cytoplasm. So remember, apical is apical surface, the top. Inclusions, secretory vesicles, and other cytoplasmic components are shed in the process. The gland cell then grows and repairs itself before it releases additional secretions. So that's how milk gets released. These just come out and it regrows that part to keep making more milk. And then we have holocrine secretion. It says down here, holocrine secretion occurs as superficial gland cells burst. So superficial, remember, it means towards the surface. So these cells up here, the cells themselves burst. Continued secretion involves the replacement of these cells through mitotic divisions of underlying stem cells. If you look at that word mitotic, I said that yesterday or on Friday in class. Turns out it is a word. I wasn't totally making that up. So if you look at the steps here about how the holocrine secretion works, you have number 1, cell division replaces lost cells. Number 2, cells from secretory products and form secretory products and increase in size. And then 3, those cells burst and release their cytoplasmic contents. So that is a sebaceous gland, and it shows you here in the picture where the sebaceous gland is. It's your oil gland. It's at the root of the hair follicle. Alright, in case you didn't see that very clearly, here it is up close, the merocrine secretion. If you want to take a look at it, go ahead and pause the video. And the apocrine secretion up close, you can see secretion happening here, and then regrowth, and then it's going to secrete itself again. And on this slide, you can see a little bit closer the holocrine secretion. Right, I hope you guys look over this as much as you want. Study your notes. Quiz coming up.